Hi, my name is Rahayel and I am presenting to you mitochondrial function-associated genes underlie cortical atrophy in prodromal synucleinopathies. Isolated REM sleep behavior disorder or isolated RBD is a sleep disorder characterized by the loss of muscle atonia and onset of movements and vocalizations during REM sleep. Most people with isolated RBD will develop a synucleinopathy, such as Parkinson's disease, dementia with Lewy bodies, or multiple system atrophy. Although still in the prodromal phase, individuals with isolated RBD already show clinical and brain changes that are reminiscent of what is seen in manifest synucleinopathies, including brain atrophy. In a paper published in Brain in 2022, we investigated the presence of brain atrophy, here cortical thinning and changes in cortical surface area, in a large T1-weighted MRI dataset of 182 isolated RBD patients confirmed with polysomnography into 161 healthy controls. We found that patients with isolated RBD had several areas with cortical thinning or increased cortical surface area. In that paper, we have shown that this pattern of cortical thinning could be recreated using a computational model called the agent-based susceptible infected removed model, which simulates the propagation of alpha-synuclein and the generation of atrophy in the brain based on gene expression and connectivity. However, this model only considered the expression of the SNCA and GBA genes. It is likely that other genes may contribute to atrophy in isolated RBD. In this work, we used imaging transcriptomics analysis and comprehensive spatial mapping to better understand the specific gene expression and connectivity underpinnings of atrophy in isolated RBD. Using the gene expression of 15,000 genes from the Allen Human Brain Atlas, the imaging transcriptomics portion of this work involved performing a partial least squares regression to identify gene expression components predicting significantly the cortical thickness or surface patterns in isolated RBD. For cortical thickness, we identified one significant component that was significant against spatial null models. A higher expression of genes negatively weighted on this component corresponded to increased cortical thinning in isolated RBD. When performing gene set enrichment and over-enrichment analyses of these genes, we found that the negatively weighted genes were enriched for functions involved in mitochondrial functions and macroautophagy. As for cortical surface area, we also identified one significant component. Since group comparisons between isolated RBD patients and controls revealed that patients had increased surface area, we focus on understanding the gene expression of increased surface area. On the significant component, increased surface area was associated with a higher expression of positively weighted genes. Gene set enrichment analysis revealed that these genes were enriched for the inflammatory response. Next, using virtual histology, we found that the genes associated with increased cortical surface area were enriched for the astrocyte, microglia, and OPC cell types, whereas no cell types gene expression was enriched in association with cortical thinning. We next performed structural and functional neighborhood analyses to know whether the patterns of thickness and surface area in isolated RBD were also influenced by the structural and functional architecture of the brain. We found that the change in thickness in a given brain region was significantly associated with a change in thickness observed in regions that were structurally or functionally connected with the region, suggesting a constraining effect of the connectome on the development of atrophy in isolated RBD. In contrast, the patterns of cortical surface area was less influenced by the structural and functional architecture of the brain, with correlations below 0.3 compared to over 0.5 for cortical thinning. When looking at the spatial mapping of the pattern of thickness change in isolated RBD over known neurotransmitter systems, resting state networks, and cytoarchitectonic classes, we found that thickness changes significantly mapped over the spatial distribution of dopamine active transporters, of the somatomotor and default mode resting state networks, and of the primary motor cytoarchitectonic class. In summary, brain atrophy is already present in isolated RBD and constrained by the brain's gene expression and connectivity patterns. Changes in cortical thickness occur more preferentially in regions with a higher expression of genes involved in mitochondrial function and macroautophagy. changes in cortical surface area occur more preferentially in regions with a higher expression of genes involved in the inflammatory response, and the structural changes in isolated RBD are also shaped by the brain's connectivity pattern, more so for thickness than surface area. Thank you.